In this Medicoapps Masterclass, we are going to learn about heme and porphyria. Now, both of these are very important topic for your PG entrance examination. A lot of questions come every year on heme and porphyria, both as a part of their biochemistry as well as, in the clinical part of the questions. So let's start, by understanding a few very important structure. First of all, you should know what is a monopyrrole. Okay, what is a porphyrin, what is a pyrrole ring, what is a monopyrrole, what is a tetrapyrrole. All these things if you understand, then the entire topic of heme and porphyrin or porphyrias will become very, very clear. So first understand what is a monopyrrole. So monopyrrole is nothing but a 5-membered ring with 1 nitrogen and 4 carbon. So this is a monopyrrole. So here you can see 1, 2, 3, 4 carbons are there and 1 nitrogen is there. So this is a monopyrrole. Is it clear? This is a monopyrrole. When 4 such monopyrroles come together, so again here is nitrogen. When 4 such monopyrroles come together, okay, and they are joined by methylene bridges. What is a methylene bridges? Carbon on one side there is a double bond and one side there is a single bond. So methylene bridges, if they are joined by methylene bridges, this is what we call as a tetrapyrrole. This is what we call as a tetrapyrrole. So one monopyrrole is nothing but you have 4 carbon and 1 nitrogen, and 4 of such rings, when they come together you will have a tetrapyrrole. Now this is also called as a porphyrin ring. The tetrapyrrole is also called as a porphyrin ring, and at the center if you add iron it will become heme for hemoglobin. At the center if you add magnesium it will become chlorophyll. So this is basically few important concepts and you didn't know how to teach it, fucker. In this Medicoapps Masterclass we are going to learn about heme and porphyria. Both of these are very important topic, and as a part of biochemistry, also as a part of clinical questions you're going to get. So before we get into what is heme, what is porphyrias, let's understand some basic structure and some basic terms. The first term is what we call as a monopyrrole, okay, or what is a pyrrole ring. So monopyrrole is nothing but a 5-membered ring with 1 nitrogen and 4 carbon. So this is a monopyrrole. You can see 4 carbon is there and 1 nitrogen is there. When 4 such monopyrroles come together, so this is one, this is your second monopyrrole, this is your third monopyrrole, and this is your 4th monopyrrole. Okay, when 4 such monopyrroles come together and they are joined by methylene bridges. What is the methylene bridges? Carbon on one side there will be a double bond, on one side there will be a single bond. Carbon on one side there will be a double bond, one side single bond. Carbon on one side double bond, one side single bond. Carbon, one side double bond, one side. So when 4 such monopyrroles are joined together by methylene bridges, that we call as a tetrapyrrole or a porphyrin. Okay, that we call as a tetrapyrrole or a porphyrin. Now this is a porphyrin, basic porphyrin ring. If you add any metal, like for example here if you add iron in the center, it will become heme. Or here if you add magnesium in the center, it will become chlorophyll. So this is basic monopyrrole, tetrapyrrole, porphyrin ring, heme, chlorophyll. So this is some very, very basic. So when we talk about porphyrin, there are 3 different types of porphyrin. So the first porphyrin is basically uroporphyrin. Uroporphyrin, so it will have 4 tetrapyrroles, 4 monopyrroles joined together, but we are talking about the side chain. So every monopyrrole will have 2 side chain. The first will be acetate, second will be propionate. Acetate, propionate, acetate, propionate, and on the fourth ring it will be propionate acetate. So this is uroporphyrin. So uroporphyrin basically a porphyrin ring, and the side chain is AP, 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 and the last one, fourth, is PA. So this is a uroporphyrin. Let's look at the next porphyrin, that is called as coproporphyrin. Now what happens? This acetate undergoes decarboxylation to form a methyl. So all the 4 acetates will undergo decarboxylation to form methyl. So this uroporphyrin to be converted into coproporphyrin, there will be a decarboxylation reaction. And now what kind of ring you have? You have got MP, 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 and the last one again will be PM. So this is coproporphyrin. The third is protoporphyrin. Now this coproporphyrin undergoes twice oxidation, two successive oxidation. Now this propionate on oxidation will become vinyl. Again, this propionate on oxidation will become vinyl. So this is how coproporphyrin is converted into protoporphyrin. So what is the ring structure? What is the side chain of protoporphyrin? M, V, M, V, and the last one will be M, P, and PM. Okay, these two are same in both coproporphyrin and protoporphyrin. Only here this propionate is converted into vinyl and this propionate is converted into vinyl. So these are the three types of porphyrin. So let's look at some important characteristic feature. The first one, if you see uroporphyrin, the word itself says uro, so it is one of the most water soluble porphyrin, and acetate and propionate are the side chain. If we talk about coproporphyrin, it has intermittent water solubility, and this acetate is converted into methyl and propionate remains the same. And last, when we talk about protoporphyrin, this protoporphyrin, basically vinyl group is there, methyl group is there, propionate group is there. This is one of the least water soluble porphyrin, and a very favorite question which we'll ask is heme is what kind of porphyrin? So heme is protoporphyrin. So in this protoporphyrin, if we add iron in the center, it will become heme. A very favorite question. Heme is what kind of porphyrin? So heme is a protoporphyrin. Okay, so once we have learned various structure of porphyrin, there are some very important points about porphyrin which you have to remember. I have told you most of these, but let's have a review of it. So what are porphyrins? These are cyclical compounds, basically 4 pyrrole rings, joined together through a methylene bridge. Okay, what is porphyrin? So we know that porphyrinogens are colorless, but porphyrins are colored. Okay, right? And there is a very important feature of this porphyrin. These porphyrin, absorb light in 400 nanometer range. So all the porphyrins will absorb light of 400 nanometer, and that will lead to a band formation. That band is called as Soret band. So this is one of the very distinguishing feature of porphyrins. They absorb specifically light in 400 nanometer, leading to the formation of Soret band. Side chain, we have already seen acetate, propionate, methyl, and vinyl, depending upon which porphyrin we are talking. Another very characteristic feature of your porphyrin is then when illuminated by ultraviolet light, they will show red fluorescence. Again, very important in diagnostic and finding of porphyrins, that when they are illuminated by ultraviolet light, they will show red fluorescence. Okay, they dissolve in mineral acid or organic acid. Now, iron porphyrin is the heme of hemoglobin. Magnesium porphyrin is the chlorophyll in the plant. Already we have done, and the three most important porphyrin, uro, copro, and protoporphyrin. So these were some very important one-liners which keep on coming on porphyrins. Having understood porphyrin, let's now move into the learning of synthesis of heme. So before we move into the synthesis of heme, can we name some of the heme containing proteins? Can you think of some heme containing proteins? So again, the proteins which will have heme is a very important topic in your MCQ examination. So hemoglobin, myoglobin, cytochrome P450, cytochrome C, catalase, tryptophan pyrrolase, and nitric oxide synthase. All of these are important heme containing proteins in our body, and you have to remember these names as a part of the examination. Now there are two very important clinical points I'm going to talk about this. First of all, any drug or any anything which induces cytochrome P450, okay, like say when you drink alcohol or when you take some drugs which will induce cytochrome P450, all those drugs will induce porphyrias. All those drugs will induce porphyria, because whenever cytochrome P450 is induced, so all those drugs in a porphyria patient can precipitate porphyria, because whenever there's an induction of cytochrome P450, which means there will be more production of heme, and if any enzyme is deficient in heme pathway, heme formation pathway, this will lead to the precipitation of porphyria. Very important point. Again, anything which induces cytochrome P450 in a porphyria patient will lead to the precipitation of porphyria. So most of these porphyria patients will come with a history of drinking alcohol. So immediately after drinking alcohol, they will start having symptoms. Second, very important, this enzyme tryptophan pyrrolase is a heme containing enzyme, and in any porphyria, because this this production is reduced, all these porphyrias may have neuropsychiatric manifestation, because this is very important in brain. And all these porphyrias, because there is a decreased production of tryptophan pyrrolase, will present with neuropsychiatric manifestation. Both these points I'll again come back when I'm going to talk about porphyria, but you have to remember as an important point, what are the heme containing proteins. Now, having understood that, let's now look at biosynthesis of heme. So where does biosynthesis of heme happens? So it happens in all the tissue, right? But 85% of the biosynthesis of heme will happen in bone marrow. One very important point is that heme synthesis does not occur in mature erythrocytes. So mature erythrocytes will not participate in heme synthesis, and the reason is that heme synthesis starts in mitochondria and ends in mitochondria. So it is partially cytoplasmic and partially mitochondrial, and we all know that mature erythrocytes will not have mitochondria. So that is the reason biosynthesis of heme cannot happen in mature erythrocytes. When we talk about another very favorite question of biosynthesis of heme is, what is the starting material for biosynthesis of heme? Succinyl CoA and glycine. When we're going to see the reactions, we'll see this is the first step, or the starting material for biosynthesis of heme. Now, the entire biosynthesis of heme actually is divided into 4 stages. First is synthesis of PBG, that is a monopyrrole. Okay, because if you see, both these are linear structure, and this linear structure is first converted into a cyclical structure. Then this monopyrrole is converted into a tetrapyrrole. This is the second stage of formation of heme. Now, this is a very important stage, because till the time tetrapyrrole is not formed, any porphyria will not have any cutaneous manifestation. Only once tetrapyrrole is formed, okay, you will start having cutaneous manifestation. Again, this point I'm going to cover again when I'm talking about porphyria. The third step is when we convert this UPG, uroporphyrin, into protoporphyrin. Okay, and the last step is basically when we add iron to this protoporphyrin, making it heme. So these are the 4 steps of synthesis of heme. So let's look at the first step. The first step is basically when succinyl CoA combines with glycine to form delta aminolevulinic acid. So this is your glycine, this is your succinyl CoA. Both of them, it's a condensation reaction, and delta aminolevulinic acid is formed. Okay. So what is delta amino? So suppose this is, because this is your acid group. Okay, so this is your alpha carbon, this is your beta carbon, this is your gamma carbon, this is your delta carbon. On the delta carbon, you have got an amino group. So this is delta aminolevulinic acid. Okay, now very important question, multiple times this has been asked, what is the cofactor for this reaction? The cofactor for this reaction is pyridoxine phosphate, or B6 vitamin. Okay, the enzyme here is ALA synthase, because ALA is being formed, and this is the rate limiting step of biosynthesis of heme. As all of you know, what is a rate limiting step? The slowest step of any cycle is the rate limiting step. So this condensation reaction, when succinyl CoA and glycine combines to form delta aminolevulinic acid, this is the rate limiting step of biosynthesis of heme. Now, there is a very important point I would want to tell you. First of all, this ALA synthase has 2 isomers. Okay, ALA synthase 1 and ALA synthase 2. And at this point, you have to remember what is the difference between these two, because again, questions are asked on this in the examination. Remember, the first difference is where they are found. ALA synthase 1 is primarily found in liver. Okay, in all the other tissue. ALA synthase 2, on the other hand, is primarily found in the erythroid precursor cells. Okay, so this is one difference where they are found. Now, the first, let's understand about ALA synthase 1. ALA synthase 1 is under feedback inhibition of heme, which means as the amount of heme increases, the ALA synthase 1 will be inhibited, and as the production of heme goes down, the activity of ALA synthase will happen. Okay, so this is very important. I will talk about the clinical significance again in a while, but remember, this ALA synthase is under the inhibitory effect of heme. So as the amount of heme increases, ALA synthase will decrease. As the amount of heme decreases, ALA synthase will increase. Whereas this ALS2, and this is very logical also, if you have increased amount of heme, you don't want this ALA synthase to be more active, you want it to be less active. Whereas this ALS2, which is found in erythroid precursor cell, this has no role, this has no feedback inhibition from heme. Actually, it is under the positive regulation of globulin. Again, this makes sense, because when you are in erythroid tissue, you just don't want to form heme, You want this heme to combine with globulin to form hemoglobin. So if the globulin is there, the heme synthesis should increase. If the globulin level falls down, the heme synthesis also should come down. So this is, it is under the positive control of globulin. So as the amount of globulin increases, the amount of ALA synthase activity will increase, formation of heme will increase. As the amount of globulin decreases, the amount of ALA synthase activity will decrease, the amount of heme will decrease. So this is very, very important in terms of what is the difference between ALA synthase 1 and ALA synthase 2. The next step is basically a dehydration step. Dehydration means removal of water. So two molecules of delta aminolevulinic acid condense and water is removed. Water is removed to form prophylinogen, also called as PBG. Okay, prophylinogen. Now, if you see this PBG, you can see that there is 1, 2, 3, 4 carbons and 1 nitrogen. So your first monopyrrole is formed. Monopyrrole ring, I have already told you what is monopyrrole. It is basically 4 carbon with 1 nitrogen, and you can see these two beautiful side chains which will decide which kind of porphyrin it is going to form. So this is your acetate and this is your propionate side chain. So is this clear? So the first PBG, and what do we call this enzyme? We call it as ALA dehydratase because water was removed at this step. So this delta aminolevulinic acid, water was removed. So with the help of ALA dehydratase, form prophylinogen. This is a very, very important step. Now another very important clinical point here is that this enzyme is primarily inhibited by lead. So in lead poisoning, this is the enzyme which is going to be inhibited. So what is the compound which will increase in delta aminolevulinic acid in lead poisoning? It is delta aminolevulinic acid because this is inhibited, this step is inhibited, so the amount of delta aminolevulinic acid will increase. Another very favorite question they will ask you is which enzyme activity is increased in lead poisoning? We know that this is decreased. So which enzyme activity is increased in ALA synthase? Now when we know that in lead poisoning, the formation of heme will come down, right? Because this enzyme is inhibited. Another very important enzyme which is inhibited by lead is ferrochelatase. There are two enzymes which are inhibited by lead poisoning. One is this dehydratase and one is the last enzyme where ferrochelatase. We'll learn about it. So ultimately what happens in lead poisoning, heme synthesis comes down, and once heme synthesis comes down, the activity of ALA synthase will increase. So this is the second question they are going to ask about lead poisoning. So lead poisoning, there are three questions, there are three questions they are going to ask. First of all, which enzyme is inhibited? Primarily this. Secondarily, ferrochelatase. Second, what is the compound which will increase? So delta aminolevulinic acid will increase. And third question is which enzyme activity is increased? So the activity of ALA synthase is increased in biosynthesis of heme. So this is the second step. Then what happens? 4 of these monopyrroles condense together, and at this step amino is removed. Okay, and then you have your first tetrapyrrole being formed. First tetrapyrrole will form. So this is basically what we call as PBG being converted into HMB, hydroxymethylbilane. Okay, hydroxymethylbilane is the first tetrapyrrole which is formed. Now remember, so there are few important points. First of all, remember, because amino is being removed, this enzyme is also called as PBG deaminase. Because amino is being removed, this is called as PBG deaminase because The second is this enzyme is also having another name. This enzyme has actually 3 names. First is PBG deaminase and second is HMB synthase. Again, very clear because HMB is being formed, so it will be called as HMB synthase. The third name for this enzyme is UPG1 synthase. We'll talk about why it is called UPG1 synthase in a while, but are you clear? These 3 enzymes, and basically the monopyrrole basically is combining into a tetrapyrrole. So PBG is converting into hydroxymethylbilane, and at this stage basically amino is being removed. The third step is when this hydroxymethylbilane is converted into UPG3. And the enzyme is UPG3 synthase. So what is the reaction which is happening in the UPG3 synthase? Okay, so we know that it is A, P, A, P, A, P, A, P. Okay, when this last bond is being formed, this AP becomes reversed. So now you have got A, P, A, P, A, P, and now it is PA. So this is what is the UPG3 synthase is doing. Okay. This is what the UPG3 synthase is doing, and now with the UPG3 synthase you are having UPG3. Now what happens, imagine this enzyme is absent. This enzyme is absent. Then hydroxymethylbilane spontaneously converts into UPG1. If this enzyme is absent, hydroxymethylbilane spontaneously converts into UPG1. Okay, and what is UPG1? Again, UPG1 is nothing but A, P, A, P, A, P, and AP. Okay, but this isomer is, not biologically useful. Okay, the UPG3 is biologically useful, UPG1 is not biologically useful. So that is why this enzyme, because it will convert PBG into UPG1 when this enzyme. So this enzyme is also called as UPG1 synthase. Hope you are clear. So the same enzyme will convert hydroxymethylbilane into UPG1, and that is why this enzyme is also called as UPG1 synthase. So three very important name of this enzyme, hope you remember: PBG deaminase, HMB synthase, and UPG1 synthase. So now let's move forward. We have seen how hydroxymethylbilane is converted into uroporphyrin 3. Now we have seen that UPG3 is going to be converted into CpG3 with the help of decarboxylase. I've already told you all the acetyl will be decarboxylated into methyl. So this is UPG3 getting converted into CpG3. Now I told you at the start of the biosynthesis of heme that biosynthesis of heme starts in mitochondria and ends in mitochondria. Very, very important. Biosynthesis of heme starts in mitochondria and ends in mitochondria. So the first step takes place in mitochondria. Then this delta aminolevulinic acid comes out of the mitochondria, and once this CpG is formed, this entire CpG is again transported to mitochondria. Now the last two steps will again take place in mitochondria. Is this clear? The first step and the last three steps will take place in mitochondria. So delta aminolevulinic acid is formed in mitochondria, it comes out, and once CpG is formed, it is again transported back to mitochondria. Again, I told you that this CpG will undergo two successive oxidation to form protoporphyrinogen A. Remember? So the first oxidase and second oxidase. First coproporphyrinogen is converted into protoporphyrinogen, and this protoporphyrinogen is converted into protoporphyrin. Okay. Then the last step is basically protoporphyrin is converted. So at this step, basically what happens, protoporphyrin is converted into heme with the help of ferrochelatase, and ferrochelatase basically iron is added. Now there are some very important points at this. First of all, remember I told you there were two enzymes which were inhibited by lead poisoning. One, and the most important, was dehydratase, and second was ferrochelatase. This is it. Second, this is the slowest step or the rate limiting step, remember? But the second most slowest step is this. The second most slowest step is this. Okay. I'll tell you the clinical significance when I'm going to talk about porphyria. Just remember this point. And third, this step is not just dependent on the activity of the ferrochelatase. This step requires iron. Okay. So this step is basically decided by the amount of iron you have got. Okay, this step is not just decided by the enzyme activity. This step is also decided by your iron status. Okay, because remember, iron is a very tightly regulated material in our body. We don't have excess of iron. So even though you may have protoporphyrin, but if you don't have enough iron, it will never be converted into heme. So all these are important points. This will become clinically significant as I'm going to talk about porphyria. Then you will understand the importance of these points. Okay, so this completes your biosynthesis of heme. Again, something very, very simple. Succinyl-CoA plus glycine will be condensed into delta aminolevulinic acid. Two molecules of this delta aminolevulinic acid, water will be removed. So it is a dehydratase. You will have one molecule of PBG. 4 molecules of this PBG amino will be removed. Deaminase, it will be converted into hydroxymethylbilane. Restructuring of this hydroxymethylbilane, AP, 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 and the last one will become PA. That then UPG3 will be formed. Decarboxylation of UPG will form CpG3. CpG3 will be transported back into the mitochondria, where two successive oxidation will lead to formation of protoporphyrinogen 9, formation of protoporphyrin 9, and finally iron will be added with the help of ferrochelatase to form heme. So this is basically biosynthesis of heme. Once we have learned biosynthesis of heme, let's learn porphyria.